This will be a short lecture to wrap up our discussion on Chapter 12, cell cycling, cell signaling pathways. And today, what we're going to be covering in particular, we've been kind of jumping around in Chapter 12, but what we're going to be covering today is Section 7, Section 8, and Section 12. Okay, Section 7 and 8 are both rather short sections. Um, in Section 12, it's a little bit longer, but these sections don't really go into a whole lot of detail. So I really won't be expecting you to know a lot of detail on these as far as an exam question goes. Any question I ask on the exam will be really just with the sort of general concepts that are covered in the book and that I sort of highlight here today. Section 7 deals with integrins. We'll talk about those first. Section 8 is nuclear hormone receptors, and we'll mention those. That'll probably be really very short when we cover this. And then the last section here, section 12, it talks about oncogenes, tumor suppressor genes, and then programmed cell death or apoptosis. And these concepts are, really important to know if you go on into, medicine or developmental biology, those sort of fields. You'll see these a lot more in future classes and in grad school. Right. When we talk about apoptosis, I'll go back to the title slide because I really like that figure a little bit better than the one that's in the book, but we'll see both. Moving on to integrins. Okay, this is from section 7. Okay, integrins are receptors on the outside of the cell membrane that interact with extracellular proteins. And those in particular, they interact with a very specific 3 amino acid sequence that's known as the RGD, and that stands for arginine glycine aspartate pattern. Okay, and some extracellular proteins that contain that are collagen, fibrinogen, and fibronectin, and there's some others as well. Those are the main ones, and some of these we've sort of mentioned before, and we will see them again. Okay, those extracellular matrix or ECM, those are the proteins of the extracellular matrix that integrins. You can think of those as sort of like a ligand for, or at least the 3 amino acid sequence in them as the ligand of the integrins, at least on the outside of the cell. Okay. When integrins react with or interact with that sequence, it triggers a cytoskeleton rearrangement inside the cell, and it can also trigger gene expression inside the cell. Okay, so that would be an example of an outside-in signal from integrins. You can also have an inside-out signal through integrins, and that happens when you have some sort of expressed gene inside the cell. It binds to the intracellular domain of the integrin. That triggers a change in the extracellular part of the integrin, and then there's an extracellular response to that. And the easiest example of that is the integrin might be in an inactive form, and then something changes. A signaling molecule binds to the inside of it, and it activates the outside part of the integrin, and then it can bind to the extracellular matrix. So this figure is not from the book. I liked it better than the one that was in the book. And we can just start here. So you can assume that the integrin, when it's folded over these extracellular domain of the integrin. When that's folded over, we kind of assume that it's closed, and that's an inactive form of it. Although when I was doing some reading about this, that was a little bit ambiguous, I think. But we'll just assume that that's the case. Okay, and there might be some inactivators stuck to the intracellular domain here that make this and lock it into the inactive form. And before we go any further, I should mention that these integrins are dimers. They have an alpha part and a beta part, and in the human genome, there are actually 18 different versions of the alpha subunit and 8 different versions of the beta subunit. And then different combinations of those give you different specificities for what they bind. Okay, so there's 18 alphas and 8 betas, and then you can, depending on what particular cell you are and what genes are being expressed, you can express, a few of these, or maybe just one specific alpha, one specific beta, and those are the ones for that are specific for different ligands. Okay, you won't need to know them in detail. That would get a little bit too complicated, but just know that there are 18 alphas and 8 betas. I think that's fine. Okay. Okay, so when this and also I should mention too that we've seen when we talked about chapter 11 and different types of membrane proteins, integrins are type 1, and that's that they have a single transmembrane helix region. So they're sort of, like a bigger part on the extracellular side, and they have some intracellular part out here, and then there's only a single transmembrane helix that connects the extracellular and intracellular. Okay, so they pass through the membrane just once in one helix. Okay, so when this is in the inactive form, a certain response inside the cell, and this would be an example of an inside-out signal, some sort of a response triggers this to be activated. So in one case, you might have the inactivators leaving by some sort of a signal. And then another important piece to this, to integrins, is this protein called talin. Okay, talin, that binds to the intracellular domain of the beta subunit that activates the integrin, and then it goes from this bent form in the extracellular part to a straight or open or active form. Okay. When it's straight like this on the extracellular part, then it can bind its ligand in the extracellular domain, and that's the ECM, extracellular matrix. Things like collagen protein, and remember that's the RGD sequence in the collagen that it can bind and adhere to. So this is an example of the way a cell can sort of clamp down on the extracellular matrix and be immobilized. Okay. You can also get some what's known as outside-in signals, where you have a change in the extracellular matrix, and you can get integrin clustering in the extracellular matrix. That triggers the intracellular domain to bind other signaling molecules like protein kinases. So these are just different kinases here. In ERK, I believe we've seen examples of in different pathways. Some of these others we haven't. You don't need to know them specifically. But what they do is then they signal a kinase cascade. Okay, things that integrins are responsible for: cell cycle regulation, organization of the cellular skeleton. Okay, what's not shown in this example is that there's a cellular skeleton that the inside domains can bind to, so you can get a response from the outside that changes the cellular skeleton, and it allows that cell to adopt, maybe become a little bit more flexible. And then also some of these signals will signal other receptors to be then put into a cell membrane so that they can pick up other signals from outside the cell. Okay, and an example of a pathway, or a specific cell type that uses integrins is platelets, and they use integrins to initiate interaction with coagulation factors in the initiation of blood clotting. Okay, so that's, I think, pretty much all that we need to talk about in integrins. There's also this, what they call inside-in signaling pathway, when the integrin becomes in a the membrane gets through endocytosis, becomes a endomembrane and like an inside vesicle. But we don't need to talk about that one. Okay. Okay. Moving on to section 8, and this is the section on hormones, steroid hormones. Okay. And it's a very short section. We'll keep it short and sweet. The information we didn't need to know about it is all really contained in this figure here. Obviously, this can get more complex if you wanted to talk about, what specific hormones there are, what genes they regulate, what genes are expressed, and what those genes do to cells. But we'll kind of keep it just in generalities here, and the general steps of this steroid hormone. I should say nuclear steroid hormone pathways. Okay. These steroid hormones tend to be very hydrophobic, and they don't necessarily flow around freely in the bloodstream. They're bound to proteins that are in the bloodstream, okay, that bind that hormone, and then those proteins can kind of hide that hydrophobic hormone inside of them. They can flow around, and then when they reach a cell that they need to drop it off at, what they do is they release it into the membrane. The hydrophobic hormone can freely diffuse through that membrane, and what's inside the cell, it'll find its way into the nucleus through the pores in the nuclear membrane. The nuclear membrane has some pretty large pores, right? Because you need to have the messenger RNA that's being transcribed in there has to get out through these pores. Okay, so there's some large pores that can diffuse through, and when it's in there, it binds to what's known as a nuclear receptor, and those are proteins that have a binding site for that hormone. When that hormone binds, usually they sort of dimerize together or form larger complexes. And then they bind to a particular region on DNA known as an HRE, or hormone response element. Okay, when they bind there, they can either, and this is sort of what's known as an effector site, and it either activates the expression of a gene that's right after that in the DNA sequence. So in this case, these receptors that have bound their hormone can bind to this HRE. When they do that, it signals the ribosome to come there and latch on, and then start the the transcription, the expression of that gene that's right after this HRE. And this, it can be a little bit complicated. Next semester, you'll probably go through and talk about some more details in gene expression. But in this semester, we can just assume that these nuclear receptors with the hormone bind to HREs, and when they do that, they either turn on gene expression or they turn it off. They can also inactivate it. Okay. And so in this case, we're showing that the gene is turned on. Okay, that gene then is expressed, so it goes from transcribed from DNA into messenger RNA. Okay, that then goes to and I mistakenly called that a ribosome. It's not. It's DNA polymerase. So that or RNA polymerase, sorry, that does the transcription. Okay, then the messenger RNA then goes outside of the nucleus to the ribosome. Okay, then from the ribosome messenger RNA, you can make a new protein, and then that protein is going to do some sort of a alter the function of the cell somehow. And this is where the complexity occurs. These hormones can bind different nuclear receptors. The nuclear receptors can bind to HREs that are in different places in the genome and turn on or off, multiple genes in a single cell, or in different cells. They might have a different effect in gene regulation in different types of cells. So the effect of these can be complicated, right? And you can have different hormones as well. So it can get a little bit. I mean, you could basically probably have a class all about these hormones, but we're just going to be focusing on this just basic general theme of these steroid signaling pathways given here. Okay, the final thing we'll talk about in chapter 12 is in section 12, and in this section talks about brings up a couple important terms. One is oncogene. Okay, an oncogene is something that when it's mutated, or the other term is also proto-oncogene. Okay, and an oncogene is something that's a mutated gene that can lead to unregulated cell growth and eventually cancer. Right? A proto-oncogene is a gene that has the ability or the possibility to turn into an oncogene. So a proto-oncogene would be something that you could think of a gene before it becomes mutated. Okay. In an average cancer cell, I've heard this statistic before. In an average cancer cell, it takes about 6 separate mutations to become cancerous. Okay, so that it's really never just a single gene that is being mutated. It's multiple genes. And one of the easiest ways to see this is in colon cancer. Colon cancer, for the most part, goes through some very specific and specific in the genes and in the timing of the genes that are mutated that lead to a cancer. And some of them are listed here. You don't have to know these. I'm not going to test on these specific names of these genes. Okay, but what happens is, in this is over time, you'll get some mutations. You'll first part of this is you get a mutation that leads to some unregulated growth, and that becomes a polyp. That polyp then, with some other specific mutations, will start growing further, and then finally you get another set of mutations, and in particular to the tumor suppressor gene p53, that becomes mutated, and then this becomes what's really defined as a cancer, and that it can metastasize and spread. And in that part of that metastasis, or one of the things that happens in metastasizing cancers that kind of leads back to what we first mentioned in the class, integrins. These integrins in metastasizing cancers can just actually, kind of let go of the extracellular matrix that they're normally attached to, and then they kind of flow to a different part of the body. And integrins are therefore a target for advanced drugs against advanced cancers, metastasis. Right. So really don't need to know a whole lot about the specifics of colon cancer. If you're interested, they're here, and they're detailed in this figure, and the book talks a little bit more about it. But the important thing to note for this is those terms. The oncogene is a gene that's mutated that can lead to unregulated cell growth. Unregulated cell growth is, really in the end of that, it's cancer. It becomes cancer. Right. There's a green box in the text. Don't really need to focus on this, but it talks about the cyclin-dependent kinases that can be involved in cancer, and that the kinase inhibitors are good molecules to target for anti-cancer targets. Okay, don't need to know those details. We mentioned this last lecture as well. The last thing that we really want to focus on in this section is apoptosis. And what apoptosis is, that's an important term to know. Apoptosis is programmed cell death. And that's a function of cells that need to die. And that cell death, it's sort of, death is a bad thing, right? But programmed cell death is actually a very good thing to happen, because it's that word programmed, it means it's supposed to happen. There's certain examples we can talk about that. Why is it good for cells to die? Well, when in development, there's certain cells that need to change into different types of cells, or that need to just die and go away, so that if you're talking about a human, the embryo can develop. The book gives the example of the cells between fingers need to die to actually make the fingers form. Okay, so that's one example of that. Okay. The other big example when apoptosis is important is when a cell is damaged. That damage can either be random mutations in the genome, or mutations in the genome caused by environmental toxins, chemicals, radiation, things like that, damage the DNA. You don't want to pass that damaged DNA or mutated DNA onto the next generation of cells. You would rather have that cell just die and be essentially tagged for removal, and not pass that damage on. Okay. Problems with apoptosis and problems with genes that regulate apoptosis are also another major source of oncogenes or involved in cancer. Okay. This is the cycle that the book gives, and there's, you have an extracellular receptor here, the TNF alpha. TNF stands for tumor necrosis factor. Okay, that shows you the necrosis factor. That's necrosis means death, or decay. So tumor necrosis factor, it's a receptor that when it binds its ligand there on the outside of the cell, that tells this cell, okay, we need to die. Okay. Not an important, other than really maybe the name to recognize. Really don't need to know details on that. There's a really sort of complex pathway with apoptosis. Um, Basically, if you want to think about it in terms, and I've seen this follow different sequences and I've seen, looking at different figures, I've seen this follow a little bit different sequence. So I don't know if this is necessarily the first thing that happens, but you can think of the first thing that happens is this protein here. It's called caspase 8. Okay. Anytime you see that term caspase, that is a protein or protein complex that is involved in apoptosis. Okay. So procaspase is an inactive form of that protein, and when it becomes active, it is then the pro prefix is taken off of it, and it's simply caspase. Okay, so procaspase is activated to form caspase 8. That then can trigger other caspases to be activated. Okay, and notice how complicated this is, right? You start at 8, and then you go on to 3, 6, 7. Okay, so don't try to focus on the numbers here. Okay. Another major thing that happens during apoptosis is the mitochondria are become leaky, right? And one of the important things that leaks out of them is cytochrome C. Cytochrome C is located between the outer membrane and the inner membrane, and that can leak out in either damaged mitochondria or mitochondria that have been signaled for apoptosis by caspase 8. Okay. When that leaks out, it can form a complex with a protein known as APAF1, and they form what's known as the apoptosome. Okay, stands for apoptosis, and osome is just a protein complex. Okay, that then binds with procaspase 9, and when that binds with cytochrome C and APAF1, you get the apoptosome, or the active form of caspase 9, which induces apoptosis. And if we go back to the title slide, that's sort of what's shown here. And I think this is the important part to remember for apoptosis, right? You have a leaky mitochondria, whether that be some sort of a damage to it internally, or age, or if it's programmed from caspase 8. However that happens, you have cytochrome C leaking out of it, binding with APAF1 to form the active it forms sort of these long structures with APAF1 that then combines with procaspase 9 to form the apoptosome. Okay, and that's usually depicted as this kind of star-looking thing, right? You have these cytochrome C, APAF1, long sort of protein complexes. They form the inner star, and then caspase 9 kind of comes on at the ends of those and extends out. That's the apoptosome. Okay, that then can basically start degrading other proteins within the cell, and that pushes apoptosis, and eventually that will signal that cell to be removed and to be digested. Okay, so that's apoptosis in a nutshell. Okay, a reminder: we have the third exam is going to be November 5th. Okay, 5 p.m. It'll be similar to the other exams, the first two exams that we've taken. The content from this lecture, again, will just be focused on, general things, not too specific. There will be at least one question that was from this lecture or from the reading. Again, not going to get not going to need to know too many of the details there. If you have questions about this, please ask in the meeting, class meeting, or email me.